Alright, this is chapter number 16, Development of Organism and Continuity of Life. We were reading about reproduction in plants, and in today's lesson we have to see all about seed, its formation, how the seed germinates, the type of seeds, the conditions which are required for seed germination, and then we will also see the types of germination. But before starting this topic, we should know what germination is. Germination is the development of a new plant from the embryo in a seed. To understand this definition, we need to know what embryo is, and for that we need to know the basic structure of a seed. Seed formation can be of different sorts since there are different sorts of seeds. So basic thing is the same that we read about in the previous lesson, that the fusion of male and female gamete give rise to a seed, which is pre-fertilization, we call it ovule. So let's see how the seed forms. The zygote, which is formed after the fusion of male and female gamete, grows repeatedly by mitosis to form an embryo. What embryo is? It consists of a plumule, which is a future shoot, while a radicle is a future root. Then we have a tissue called cotyledons, which stores food which is needed for the germination of the seed. So you see that the zygote, which was formed after the fusion of male and female gamete, gave rise to embryo by repeated mitosis. This embryo in turn consisted of three parts: plumule, radicle, and cotyledons. Plumule is the growing shoot, while radicle is a growing root. Cotyledon provides the food which is stored in it for the seed to use to germinate. Here in the diagram, you can see how the endosperm nucleus is formed by the fusion of one male gamete with the definitive nucleus or polar bodies, and the other male nucleus or the male gamete fused with the ovum or the egg in the ovary, giving rise to a diploid zygote. Here again, I have tried to show you how the endosperm is here to store food for the developing embryo. The green part is showing you cells of the embryo. Among these cells, some will form plumule, which is the young shoot or the future shoot, while the radicle is the growing root. Cotyledons, on the other hand, would store the food for germination of the seed. Integuments that we read in our previous lessons were the layers of cells surrounding the ovule will become seed coat or testa. Endospermic seed formation includes the endosperm nucleus dividing repeatedly to form endosperm in endospermic seeds. This endosperm acts as a store food for the developing seed, for example in maize. So in maize we see endosperm, which is present there as a storing tissue. It stores starch in it so that the growing seed can use that starch to germinate or to grow. In a non-endospermic seed, on the other hand, this endosperm is completely used up in early stage of seed development, so that the stored food is only present in another structure called cotyledons. So in our syllabus, we have to focus on non-endospermic seed, where endosperm tissue is completely or partially used by the growing embryo for its germination, and the stored food would be located in structures called cotyledons, not endosperm. In endospermic seeds, we will find endosperm, while in non-endospermic seed, we will find cotyledons, such as in the bean. Here in the diagram, I have tried to show you that all the seeds will consist of embryo. This embryo consists of plumule, which is a growing shoot. Radicle is a growing root. Cotyledon would supply food for the seed leaves. All the seeds will contain embryo, while endosperm will be present only in endospermic seeds, which will store food. It will be absent in non-endospermic seeds, where we will find cotyledons, not the endosperm. Monocot and dicot seeds are drawn here, showing you that the dicot embryos develop two cotyledons, while monocot embryos develop only a single cotyledon. So you see in the diagram I have shown you two seeds. One is a dicot seed while the other one is a monocot seed, which ensures that there are two cotyledons in this seed and only one cotyledon in the corn seed. This is the basic difference between the monocot and dicot seeds. Obviously, there are some other differences between monocotyledon and dicotyledon plants, which is beyond the scope of our syllabus. So we will just focus on this difference, that a monocot seed will have one cotyledon while a dicot seed will have two cotyledons. Mind it that cotyledons are structures which are storing food in the form of starch. Here in the structure of the seed, you can see in a bean, we can see there is a seed coat, so it is present in the maize seed. Then we have a plumule, which is the growing shoot. Both are labeled. Radicle is a growing root. Cotyledon can be seen in bean and as well as in maize, where it is single. Endosperm is present in endospermic seed while it is absent in non-endospermic seed, which is the bean. If you will see the outer structure of a seed, you will find it to be a bean shape or kidney shape. There would be a hole here, which is sometimes not seen with the naked eye, but most of the times it can be seen. This is called micropyle. If you remember our last lecture, we saw what micropyle was. It was a hole through which the pollen tube was going inside the embryo sac, where it was releasing the male gametes. So this micropyle hasn't changed after fertilization. It's still there. It's still a hole. While hilum is a place where this seed was attached to the ovary wall. Seed coat or testa was called integuments before the fertilization process. We will read about the function of micropyle in the seed in our coming slides. We will also see that what is the function of seed coat or testa. For now, just remember the structure of the seed, which is a bean shape or kidney shape. We can see micropyle, hilum and seed coat or testa. In this diagram, I have shown you the internal structure of the seed, where you can see cotyledon, which is storing starch. We can see seed coat or testa. We can see radicle and plumule. The characteristic of these two and seed coat, I have shown you before, that plumule will grow into a shoot while radicle will grow into a root. These three, plumule, radicle and cotyledon, are named as embryo. If you can see here in the diagram that the cotyledons are storing starch, this starch would be converted to maltose and glucose by the help of the respective enzymes which are present in the seed. Mind it that this is happening, this digestion is happening in order to let the seed respire so that it can produce ATP, which will be used for seed germination or growth. So these steps or these reactions are obviously enzyme catalyzed, so these will require water and they will require a optimum temperature from by which they can work. The enzymes work at optimum temperature. We all know that from our basic primary knowledge of enzymes. And also remember, since respiration is happening, so the seed would also require oxygen. Here in the diagram, in the table, I have placed the functions of the structure of the embryo. Plumule is growing shoot, as I've told you before. The radicle is a growing root of the new plant. Cotyledon would store food, where the digestion of the stored food would occur. Testa is a hard outer protective covering. So seed coat is actually protecting the seed. Micropyle, on the other hand, is a hole through which water and oxygen will enter and carbon dioxide would leave. If you see the previous slide, I told you that the starch is converted to maltose and glucose by the respective enzyme, that is amylase and maltase. Once the glucose is formed, this will be used in respiration, which requires oxygen and liberates or release carbon dioxide. So micropyle is not only involved in the absorption of water but also for the incoming of oxygen and exit of carbon dioxide from the seed. This ATP energy then will be used by the seed in the process of germination. Here again in the diagram I have shown you the basic structure of a seed. This is the seed coat. Plumule, hypocotyl is the base of the radicle. Radicle is the growing shoot. Radicle is the growing root. Cotyledon is the tissue which is storing the food in the form of starch. Epicotyl is the base of the plumule. Embryo consists of radicle, plumule, and cotyledons. This is the process of germination where you can see that the radicle is growing first and downward. Then the shoot is growing upward in upward direction. Keep in mind that the radicle will come out first because it has to absorb water and mineral salts, and also it has to anchor the seedling into the soil. So the radicle would move downward and will be first to grow from the seed. Then the growing shoot will appear going upwards towards the upper part of the soil in the atmosphere, so that it can grow leaves and start doing photosynthesis. So till the seed is inside the soil, it is dependent upon the food which is stored in the seed in the form of starch. But once the shoot is gone upward into the atmosphere, it is able to produce leaves which will be having chlorophyll and will be able to do photosynthesis for itself. Now the seedling then won't be dependent upon the starch which is stored in the seed. Here in this diagram also I've tried to show you how the radicle is growing first and in downward direction, while the shoot is going up, and then the cotyledons are giving rise to leaves. Leaves are green in color, having chlorophyll, and are able to do photosynthesis. Once they are able to do photosynthesis, they will no longer be dependent upon the stored starch in the cotyledon. If you can see in this animation, you can see that the cotyledons are coming out of the soil. Such kind of a germination where cotyledons appear outside the soil, they come out of the soil, such germination is called epigeal germination. Here you can see it again in the animation that the cotyledons are coming out from the soil. It does not happen every time with every seed. This is happening with the bean seed though, that the cotyledons are coming out of the soil. So this germination is called epigeal germination, where the cotyledons come out of the soil. So now we have to see what are the conditions which are required for seed germination. We read that seed germination requires to have ATP, and for that the seed will undergo respiration of respiring the glucose which is formed from the starch which was stored in it. If we provide water and warmth to our seeds, the seed coat will absorb water and eventually crack. So now we know that the water is the basic requirement for seed germination. Water is required because it has to soften the testa so that the growing shoot and root can come out. So if you have observed in our common practice, you would have observed that before sowing the seeds into the soil, we sometimes place them in water before so that the testa gets softened and cracks. So when we sow it in the soil, it is easier for plumule and radicle to grow outward. So the first condition which is required for seed germination is water, because water is required to soften the testa so that the growing root and growing shoot, that is plumule and radicle, can come out of the seed. Once the seed coat opens, the seed will consume food from the cotyledon and begin to grow. So first condition was water. The function of water is not only to soften the testa, but also to activate the enzymes which are present in the seed. If you remember, we know that enzymes work in aqueous medium. So water is not only required to soften the testa to let plumule and radicle come out, but it is also used to activate the enzymes which are present in the seed. The other condition is suitable temperature, for we know that for optimum activity of the enzyme we need to have a suitable temperature. So the second condition required for germination is temperature. Then we have oxygen, which we have already discussed that it is required for the process of respiration, so that the stored starch is converted to glucose ultimately by the help of the enzyme, and that glucose must be respired or oxidized to produce ATP. This process would require oxygen. So these are the three basic conditions which are required for germination. Here in this diagram, I have elaborated the difference between epigeal and hypogeal germination. I told you that epigeal germination is that one in which the cotyledons come out of the soil, while hypogeal germination involves where the cotyledon stays inside the soil. It does not come out with the seedling.